My name is Kristen Harmon. Today I'm going to be talking to you about the first year of my master's thesis research entitled Evaluating Impacts of Nest Initiation Time and Nest Site Location on Hawaiian Stilt Nesting Success in Windward, Leeward, and North Shore wetlands on Oahu. It's a mouthful. So the Hawaiian stilt, or Io, is a federally endangered native Hawaiian waterbird. It's also a subspecies of the blackneck stilt. You can find Hawaiian stilts in wetlands throughout the Hawaiian Islands, but you can also find them in golf courses or wastewater treatment plants, pretty much anywhere there's shallow water or mud flats. The Hawaiian stilt nesting period is from mid-February to about August. And as you can see from these pictures here, the Hawaiian stilt has very distinct morphological characteristics. So some of you may have actually seen them before, but if you haven't, take note to their really long, skinny, pointy bills, as well as their really long, skinny, pink legs. They're called the Hawaiian stilt for a very good reason, as they look like they are walking on stilts. The nesting habitat of Hawaiian stilts greatly varies. Some of their nests can be constructed from vegetation. Some can be constructed from sticks or stones. Actually, I've just recently observed some that actually use little seashells. It's really cute. And they kind of decorate their nests with those. And then some are really built up. So this picture in the top left corner is a built-up nest, whereas some of these other nests are more low-lying and simple. They can also be placed in dense vegetation, like this one in the bottom left, or they could be more exposed, like the pictures on the right-hand side. The clutch size of the Hawaiian stilt, meaning the number of eggs, varies from 1 egg to 4 eggs, but on average has about 4 eggs. Because the Hawaiian stilt is a ground nesting waterbird, it is highly susceptible to flooding and predation. Flooding can occur in a couple of ways. One way is that the surface water levels will rise in the wetland enough that it completely covers the eggs, as you see in this picture on the left. Another way is that the surface water levels will rise very rapidly, and this can actually wash the eggs out of the nest as well. Another threat to nesting success is predation. And this picture on the right is a picture of a predated stilt egg that I found presumably predated by a mongoose. And unfortunately, the Hawaiian stilt has a number of potential predators. These include native predators, but the majority of them are non-native. Some of the predators are avian species, such as the black-crowned night heron, barn owls, cattle egrets, potentially even pueo, which is the Hawaiian short-eared owl. Their predators could also be aquatic, more like the bullfrog or catfish. And then they also have potential land mammal predators, such as feral cats, rats, and mongoose. And there could be potentially other predators that I have not listed, but these predators I've mentioned are considered the main potential predators for the Hawaiian stilt. And a number of previous studies have actually identified a correlation between surface water levels and nesting success for other waterbirds. For instance, high surface water levels make nests more likely to be flooded, but they can also act as a deterrent or a barrier to nests, as they make nests less accessible to land predators. So that could also decrease predator diversity. Conversely, low surface water levels may make nests less likely to be flooded. However, they could also make nests more accessible and exposed and easily accessed by a number of different predator types. Another thing that could happen with lower surface water levels is the wetlands where their nesting could dry out. which could impact food availability, which may lead to nest abandonment or even death of the chicks if they've hatched already. So these previous studies that have been done on other water birds, they haven't been done on the Hawaiian stilt. And one of the reasons why this relationship between surface water levels and nesting success would be important to look at here in Hawaii is because precipitation, as we all probably know, varies spatially and temporally here in Hawaii. So not only do we have a wet and dry season, but we also have wet and dry sides of the islands. And so this leads to varying hydrological conditions of the wetlands. For instance, some wetlands may be drier or wetter depending on which side of the island they're located on and what time of the year it is. Therefore, causes of nest failure may also vary spatially and temporally in wetlands across the Hawaiian Islands. So for my first year of research, I wanted to kind of do a preliminary study since there really isn't a whole lot of information on Hawaiian stilt nesting success. I wanted to specifically identify the causes of Hawaiian stilt nest failure and as well as when and where they occur. I also wanted to examine the relationship between the cause of nest failure, such as predation or flooding, and nest site location, as well as nest initiation time. So when I'm referring to nest site location, I specifically mean the different locations on the island. From my study, that includes the North Shore, the leeward side, and the windward side. And nest initiation time, I'm specifically referring to the date or month in which the Hawaiian stilt lays the egg. So that's considered when they initiate the nest. As I mentioned, my study sites are across Oahu. They include James Campbell National Wildlife Refuge, which is on the north shore of Oahu, Pearl Harbor National Wildlife Refuge on the leeward side of Oahu, and Kauainui Hamakua Marsh Complex, which is on the windward side of Oahu. And within each of these nest site locations, I have chosen 2 wetlands to do my nest surveys. On the windward side, I am doing surveys in Kauainui Marsh and Hamakua Marsh, which are located in Kailua. Respectively, they are on top is Hamakua Marsh, and on the bottom is Kauainui Marsh. And in Kauainui Marsh, I am specifically studying these ponds right here, which are about 40 acres of the 800-acre wetland. So just a smaller portion of the wetland. On the leeward side, I am doing surveys in Waiava and Honouliuli wetlands, which are located in Pearl City and Waipahu, respectively. And the top picture is a picture of Waiava wetland, and the bottom is Honouliuli wetlands. So as I'm going through these pictures, you can see there's great variability between the wetlands. So some have mud flats, some are a little drier or wetter, and all have different types of vegetation as well. And on the north shore of Oahu, I am doing my surveys at James Campbell, and specifically I am doing my nesting surveys. Up top is a picture of the ponds in the Ki'i unit of James Campbell, and the bottom picture is a picture of Pueo Flats, which is a large mud flats that the stilts enjoy nesting on. I conducted my first preliminary research from February to August of last year. I identified breeding pairs in every wetland, and once I observed that breeding started, I started my nest searches. I was not able last year to completely go into the wetland and search all through by foot. So what I did was observational surveys from around the perimeter of the wetlands. Most of these wetlands have easy access roads that you can use to observe and do surveys. So I used a spotting scope and binoculars to identify the pairs as well as the nests. Then I recorded the approximate location of each nest that I located, as well as the nest site characteristics. So things like vegetation type, vegetation cover, water levels, those kinds of habitat characteristics. And this picture of the stilt that's sitting, sometimes it was easiest for me to wait and observe a sitting stilt. That possibly means they're sitting on a nest. So I would wait, and sure enough, this stilt here was sitting on a nest. I waited till it left the nest, and then I was able to observe how many eggs were inside. And then for my data analysis, I classified nest failures as either predated, flooded, abandoned, or unknown. Sometimes it was difficult to tell what had happened to the nest, so I did classify it as unknown. And then I quantified the nest failures due to each type, either predation, flooding, or abandonment. And for my known causes of nest failure, for each, I ended up binning the nest initiation times into months. So I'm specifically looking at the nest initiation months of April, May, and June, which were all of the months in which nests were initiated. And for each of those nest initiation months, I calculated the hatching success for the windward, leeward, and north shore wetlands so that I may compare them. And lastly, I did a statistical analysis to determine if there was a relationship between the cause of nest failure and nest site location, as well as nest initiation time. So taking a look at some of my preliminary results, I did identify, my statistical test did come back with a significant relationship between cause of nest failure and nest site location and nest initiation time. And if we look at the breakdown of hatching success, this graph shows hatching success in each region of Oahu for each nest initiation month. So a couple of things I want to point out is that we do see that the leeward side had higher hatching success in all of the months. And then we also note that for each nest site location, the hatching success increased with nest initiation month. So it seemed that as the season progressed, the hatching success also increased. And this graph is basically a summary of all of the causes of nest failure that I found and when and where they occurred. Actually, this is actually showing for April, May, and June, all of the months. This is still nest initiation month. This is not when the nest failed. This is saying that for April, the nests that were initiated in April, this is what happened, their nest fate. This is what happened to them. So there's a lot going on in this graph, but I'll just point out a couple of interesting things. The first being that for the windward wetlands, I did observe all causes of nest failure in that location, whereas the leeward wetlands, I only observed predation as a cause of nest failure, and in north shore wetlands, I only observed abandonment as cause of nest failure. For abandoned nests, just to clarify, I consider a nest abandoned if I observe the nest still has eggs inside and it's well past the expected hatch date, in combination with the fact that there are no stilts present. So stilts are actually very territorial and defensive of their nests. And so it's very odd to approach a nest and not have anyone screaming at you or trying to hit you. So for these abandoned nests, I specifically determined that they were abandoned by seeing that there were eggs inside past the expected hatch date and there were no stilts present nearby. So that was my criteria for determining abandonment. Another thing to point out is that the flooding events, which are in blue, I only observed flooding of nests in the windward wetlands and in the month of April, or for nests that were initiated in the month of April, which is kind of what we would expect since the windward wetlands on the windward side of Oahu has the most variable rainfall and potentially the most variable surface water levels. So now we'll actually look at when and where the nest failures occurred. So this first graph is just of my flooded nests, and this is the rainfall for the windward side of Oahu, specifically in Kailua. And as you will see, as we would expect, nest failures due to flooding occurred either when there was an increase in rainfall or shortly thereafter. I did want to point out that the stars are representative of when I found the nest. So the flooding could have occurred a couple of days prior, since I wasn't there every single day in that wetland. For abandonment, I did observe a couple of nests abandoned in Hamakua Marsh on the windward side. However, for this particular graph, I just chose to show the North Shore wetlands because that was where the majority of the abandonment happened for this season. And when you look at this graph, you see that I observed abandoned nests either when there is fairly low or stable rainfall or during a decrease in rainfall. And for predation, I did find predated nests in both windward and leeward wetlands. And this first graph is of predation events that I observed in the windward wetlands. And as you see, they all occurred either when rainfall is very low or stable. But it's interesting to compare this to the leeward wetlands because all of the predated nests that I found in the leeward wetlands were actually after an increase in rainfall. So that's kind of an interesting result. So in summary, I kind of threw a lot of graphs at you. So just to summarize, some of the key things I found for my first year were that for all nest sites, hatching success increased as nest initiation time increased. Hatching success was lower for all of these wetlands earlier in the nesting season, which could be due to a few things, possibly the first being that there is more variable rainfall in the early months of the nesting season, since we are still technically in our rainy season. We're coming out of that. But also could be just because there were less total nests altogether. So the peak nesting season for stilts is known to be May or June. And so there's just not as many nests in the first part of the nesting season. Abandonment occurred later in the breeding season, which could be related to the drying out of wetlands. Unfortunately for Pueo Flats, I did not have a staff gauge, which would allow me to read the water level every time I go, but I was able to visibly confirm that the wetlands there did dry out completely during the same period that I observed the abandoned nests. So my thoughts for that are that potentially, as I mentioned before, drying out of wetlands can affect food availability, but abandonment of nests could also be due to predation pressure. So if the stilts are seeing a lot of potential predators around, as I mentioned before, water levels being low could make the nests more accessible to predators, which would make them come out more and be more visible. So abandonment could also be caused just from predation pressure. And for the predated nests, I actually noticed that they occurred throughout the nesting season and in all wetlands. So there wasn't a particular time or place where they occurred. And so all of the relationships, these relationships I've mentioned, need to be evaluated further, which is why the next steps of my research, I'm now in my second field season, and this time I'm actually able to exhaust the wetlands looking for all of the nests possible. So you probably noticed that some of my sample sizes were pretty low for the first season. Again, that's because I wasn't able to exhaust the wetland completely for all of the nests. So this season, I'm currently doing that. I'm also using nest cameras so that I can identify top predator sources. We do know there's a variety of potential predators, but we need to identify what the top predators are to the stilts. I also want to look at the threshold in rainfall in which each wetland site becomes flooded. So every wetland has different hydrology, and they're not all going to require the same amount of rainfall in order to become flooded. So it's important that we look at that for each wetland. And so I'd like to develop, I'm actually working on a model at the moment that would let me know, given certain rainfall or stream flow, what depths of flooding the wetland would endure. I'm also going to use that model to predict the rate of evaporation following rainfall. So again, not all wetlands are going to dry out at the same rate. So we need to identify that as well. And lastly, I'm going to statistically evaluate the relationship between rainfall intensity, surface water levels, and causes of nest failure due to either flooding or predation. So now I'm going to actually look at that relationship. So I have those graphs of when and where the nest failures occur, but I need to statistically evaluate them and create models for that so that we can possibly predict in the future, given certain rainfall and certain water levels, what's the likelihood of these nests becoming flooded or predated. So with that, I just want to quickly acknowledge my lab, Hawaii Wildlife Ecology Lab. There are a number of people in our lab that have helped me conduct my research, as well as taken beautiful photos for me. And so I thank those people as well as my committee members. As I said, I'm in a master's, so my committee is providing me a lot of insightful feedback. And then I also have a number of partners that are government agencies that I am working with in order to make this research possible. So I want to thank them. And with that, I'm going to introduce my lab mate, Carmen Antaky.
Thanks, Kristen. That was an awesome talk. And also, thank you for the introduction. So I am Carmen Antaky, and I'm also a graduate student. Can you hear me? In the Hawaii Wildlife Ecology Lab, and I'm also doing my master's. I'm graduating this semester, so I'm going to share with you some of the work that I've been doing for the last couple of years, and I'm excited to talk to you guys all about seabirds, so thanks for coming tonight. As you know, seabirds are marine predators that help reveal the health of our oceans, and they are also native to the Hawaiian Islands. So they were some of the first to call Hawaii home and bring essential nutrients like nitrogen to our soils to help facilitate the growth of our native forest. But they're also spatially complex. They have large ranges and utilize both the ocean and land throughout their life. And because of this, they run into a lot of threats. So as a taxa, seabirds are globally threatened, and in particular in the Hawaiian Islands, we have a lot of endangered seabirds. And some of these, as Kristen talked about, some of the threats. And so as conservationists, we kind of have to focus on the threats. So what's really harming these populations? And for seabirds, it is also introduced predators. So they've evolved without mammalian predators. And so they're very vulnerable to cats and rats because these species can easily come and predate ground nesting seabirds that are completely vulnerable. There's a bunch of other threats from human impact, which include habitat loss, light pollution, and then more threats on the ocean, which include marine debris, which can result in entanglement, as well as ingestion, which both can lead to mortality within seabirds. So in my research, I explore phylopatric behavior within seabirds and also look at patterns of genetic diversity, particularly in one endangered seabird that nests on the Hawaiian Islands. And so, as you know, seabirds are colonial. Over 98% of seabirds nest in colonies. This is a Laysan albatross colony on Midway Atoll, if you're familiar. And many of these seabirds that nest in colonies exhibit natal phylopatry. And that's what I'm going to focus on today. And that's the behavior of a seabird returning to the colony of birth to breed. And although seabirds have a high capacity of dispersal, they can fly really great distances. A lot of seabirds are surprisingly phylopatric. So they return right back to the place they were born to lay their chicks for generations. And this phenomenon has been coined the seabird paradox. So within a life cycle of a seabird, once they fledge, they spend some time on the open ocean, sometimes up to several years, before returning to land to breed. And so what goes into making this choice? if they return to their natal colony, it usually guarantees mates and resources and usually safety. But it has the disadvantages of potentially inbreeding. There's more seabirds, so there's more competition, as well as potential ecological traps. So if the habitat has since been degraded. If seabirds go to a new colony to breed, the advantages, in contrast, are increased gene flow, lowered competition, but it's also a risky behavior because they're entering the unknown and they're unsure if this colony is safe. So this comes with the risk of mortality. And so in the past, studies have found that there is a variation in phylopatry across different seabirds. So some have really high phylopatric rates and others do not. And so I was curious to see if this was driven completely by colony structure or other factors of dispersal. And so if phylopatry is related to colony structure, you might see this linear relationship between the two. And when I mean percent phylopatry, I'm talking about the number of seabirds that return back to their birth site within 20 kilometers over all of the seabirds that we look at. And then colony size percentage is the population size of a colony over the total population size across all colonies. So if 40% of species of seabird nests on one colony, you'd expect there just to be a 40% return just by proportion. So we might see this linear relationship. But if the seabird paradox is true, then we would see consistent high returns no matter the relative colony size. Or it could be completely random, with seabirds returning at different rates no matter what the colony size percentage and averaging about a 50% return. So along with colony size, I also wanted to look at other factors that might be driving dispersal. And so this includes the number of colonies, distance between colonies, and geographic region. And there may be other species level factors driving phylopatry as well. So I looked at taxonomic order of the seabird, as well as wing load, which has been linked to the ability to fly. So that's the mass over the wing surface area of the bird. And I also looked at foraging strategy. And most seabirds can fall into two categories, non-central and central foragers. Central foragers meaning they forage in their nearshore resources close to their colony, and non-central meaning that they forage great distances away from their colony. And in this study, we used 200 kilometers away as a non-central forager. And those non-central foragers are more likely to return to their natal colony because they're less prone to local food shifts. And so I first collected the banding data from the Bird Banding Lab in the British Trust of Ornithology, and then matched that with available census data to look at the colony sizes, and then assign those different species and colony level factors I just talked about, and then applied a linear mixed effects model, which helps control for species bias when we test the model across all different kinds of seabirds. And so the areas of overlap resulted in the temperate region of the British Isles, as well as the tropical region of Hawaii. And 36 different species were included in this study. They're across 4 different orders. And because there was only one chalkbrook bird, I took that order out of the final analysis by taxonomic order. And so I just want to give you a brief refresher of what kind of trends we might be seeing when I show you the results. So they might have this linear relationship, or they might see just consistent high returns, or it might be completely variable. And so this is what we get when we look across all the 36 different seabirds. And it kind of looks like the random graph. There's a lot of variation, but you see this slight trend towards colony size. And that's significant. I also want to point out that I did log transform the colony size data because to meet the assumptions of normal distribution, so the data would lie evenly across the x-axis. Also want to point out that I got an R-square valued of almost 0.5, which shows the amount of variation explained by the independent variables. So because dispersal is this really complex behavior, 50% actually is a pretty strong number to show that as it explains what we're seeing and what trends. But it gets a little bit more interesting when we look at the data by taxonomic order. So the orange line here is the Procellariiforms. So those are the albatross, the shearwaters, and petrels. And they are consistent with the seabird paradox because they have high return rates. You see it averages over 60% no matter the colony size percentage. In contrast, the other lines, so the white line is the Chargiforms, which are the gulls, terns, and auks, and then the Suliiforms are the boobies and gannets, and they both show more of that random distribution. They average about 50% and have a lot of variability. It's also important to point out there's no p-value for the Chargiforms because they were used as the baseline. So the Suliiforms do not differ from the Chargiforms, but the Procellariiforms are significantly different in their phylopatric patterns. Other significant results that we found was that foraging strategy. So those non-central foragers were more phylopatric than central foragers, as well as the tropical region. So seabirds from tropical colonies were more phylopatric than those from temperate. And so what did we find out about the seabird paradox? We found out that Procellariiforms were consistent with the seabird paradox. They returned back to their natal colony more often than by just chance. But the Chargiforms and the Suliiforms did not. So they had no evidence for the seabird paradox. It's also important to point out that even though it wasn't a linear relationship, colony size did have a positive effect on phylopatry. And another interesting result that I found from my study is that even looking over 36 different species, not a single species was 100% phylopatric. So some dispersal is happening in some capacity within every seabird that we looked at. And so the Procellariiforms are albatross, shearwaters, and petrels, and they were phylopatric. And this is not very surprising because they're historically phylopatric and have had the high success in seabird conservation programs. So, because if you translocate a Procellariiform, they're more likely to return to that colony that they fledged from and then reap the benefits of that protected area and thus have a successful conservation for the future. But they also may be driving some of our other significant results because Procellariiforms are in this study were all non-central foragers. They forage long distances, and also most of them were found in the tropical region in this study. There was only 2 in the temperate region, and one of them, the northern fulmar, had really low rates of phylopatry because of their recent expansion in the British Isles over the last 100 years. And so we did our best to test dispersal with the available banding data and movement data that we had, but there may be other things that are affecting it that we just weren't able to test for in this study. And these include the presence of other seabirds, predator abundance, sex bias, and food availability. And so due to phylopatric tendencies, the Procellariiforms are vulnerable to ecological traps because if they consistently return to that birth colony and that colony has been degraded, that's going to affect them and threaten them in the future, and which is why we see globally that Procellariiforms are endangered. But it also means that they're good candidates for translocation. So if we get them to a protected area, they'll stay there and they can reap the benefits from the conservation. And so the next part of my study, I continue to look at phylopatry, and I look at its effects on population genetics. And so phylopatry, as I've explained, limits dispersal and thus gene flow. So it presents this conundrum for genetic diversity. And I test this within an endangered Procellariiform in Hawaii, and I also look at its effects on Procellariiforms, which have known to be phylopatric, like I just showed in the last study, and compared to Chargiforms, which are not phylopatric. And so the seabird that I studied was the bandrump storm petrel, which is native to the main Hawaiian Islands. It's actually the smallest and rarest seabird here that nests here. And we don't actually know the exact population size because we only know of where 2 confirmed nests are on all of the Hawaiian Islands, but we believe it to be very low. And we also believe that that they're threatened by many of the same things as other seabirds, and that includes, overfishing, marine debris, habitat loss, climate change, light pollution, as well as introduced predators, which I talked about before. But there has been, because they're endangered, a new push for management. So in the areas where we know that they are, there has been some predator control, and there's also possible migration between the Hawaiian Islands that may be helping to recover this species. And so my first objective was to look at the genetic diversity that's currently in the populations of the endangered bandrump storm petrel in Hawaii, and also develop more genetic resources for this bird. I was the first study to look at their population genetics in Hawaii, and so I developed microsatellite markers, which are highly polymorphic areas of the genome, which are useful for population level studies. And even though this bird is only endangered in Hawaii, it actually has a global distribution. And so this includes different island archipelagos in both the Pacific and Atlantic Oceans. And I wanted to compare the genetic structure we find in Hawaii to those globally. And I also wanted to look at other published seabird population genetic studies to compare genetic diversity between those 2 orders that are phylopatric and not. And so what I expected was to see low genetic diversity and gene flow in the Hawaiian Islands because of their endangered status and low population size. I also thought that the Hawaiian populations will be genetically distinct because they're isolated from the other global populations. And I also believe that the procelariforms would have lower genetic diversity than the chargiforms based on their phylopatric tendency and which limits dispersal. So the areas where I received samples for the bandrump storm petrel were on Kauai and the Big Island. So these are the 2 known areas of where they breed, as well as management. So the Kauai Endangered Seabird Recovery Project on Kauai sent me samples that they collected on misnetting events, and those included blood samples. And then on the Big Island, the birds are found on the Pohakaloa training area, so their team out there sent me feather samples that they found using dog searches and personnel searches. And so once I received the samples, I extracted the DNA, and then I quantified each pool of DNA of how much was in each, and I pooled them equally into 2 pools, one for Kauai and one for the Big Island, which I then was able to digest with restriction enzymes and put the size-selected fragments through the Illumina sequencer, which is a next-generation sequencing. And then I received the genetic sequences to perform bioinformatics. And so bioinformatics is the genetic analysis of DNA, and bear with me because probably not everyone's a geneticist in here, but this is where I spent the majority of my master's, was looking at a computer screen at genetic code and analyzing it. You get millions of reads from this data, and you need to have very comprehensive pipelines. You can't just stick it in Excel. You got to use pretty sophisticated coding and packages. And I won't get into all of these, but basically these are names of a few of the programs I use to compare the genetic diversity within the populations of Hawaii, and also globally compare them to the other data that we had already published and available from those samples globally. And the other part of my study was to look at the different population genetics population studies for seabirds, and I did a literature search, and I limited it to at least 2 populations, and that they had to report the average nucleotide diversity, which is a good genetic diversity estimate, which looks at the polymorphism of nucleotides within a population. And I found that most of the studies that have been done in the past did use mitochondrial sequences, and specifically the control region and cytochrome B. So those were the ones that I kept in my analysis to try to limit the different mutation rates by genes to have an even comparison. And then I used a t-test in R to look at if there was any difference between the procelariforms and the chargiforms in their genetic diversity. And so from the next generation sequencing data, like I said, I got back to about 10 million paired end reads, and I did a SNP analysis, which is a single nucleotide polymorphism. So those are the different nucleotides within a genetic code that you're able to assess the diversity of populations and species. And from that, I got about 1,500 SNPs with a 15 depth coverage. And between the two islands, I found a FST value of 0.077. So an FST value is a fixation index, which looks at population differentiation. It ranges from 0, which means that they're completely mixing, and then 1 is that they're completely different. So a 0.077 is pretty low, like it means that there's some minimal differences, but anything less than 0.1 means that there is moderate gene flow between these populations. I did see, like I said, some minimal differences, about 24 fixed SNPs, so that might help identify where individuals from which population, but they did have moderate levels of gene flow. And so bear with me when I get through some of this genetic analysis. So what I found for both of the populations using my next generation sequences was and I also looked at the mitochondrial DNA. So these are more polymorphic than ones that are the genomic DNA, which was randomly assessed across the genome. And I found that there was a negative to GMAS-D, which shows that there is an excess of rare alleles, which possibly is due to expansion or selective sweep. In this case, it's most likely because of their high mutation rate. And I also found about equal levels of nucleotide diversity between the two populations, but a higher population mutation rate, which is the Waterson's estimator, for the Kauai population. And also, when I looked at both populations combined for their mitochondrial DNA, I found that they all had the same trend, but in higher rates. And this is consistent with other studies because, like I said, mitochondrial data is more polymorphic, it's more different. So you're expected to see like higher nucleotide diversity, higher population rate, more extreme to GMAS-D. But it also shows that these populations do have relatively high amounts of genetic diversity than what we would expect for an endangered population. And I also found different microsatellites that went through high qualities of filtering, which includes like optimal base pairs, optimal melting point. And these I didn't analyze or score, but I developed for further studies that will be useful when we're adding more samples for this species. And then I also created a phylogenetic tree, which shows the relationships, the genetic relationships between those in Hawaii and others globally. And so basically this shows that there's a clustering of the Galapagos and Japan. And because Hawaii doesn't cluster with them, and those are their nearest Pacific Ocean neighbors, it shows that Hawaii looks distinct genetically from those globally. I also found 44 different seabird population genetic studies, 21 from the procelariforms and 23 from the chargiforms, but I found no significant differences in nucleotide diversity. There was a lot of variation within both orders. And so that suggests that there's other mechanisms that are influencing this, this variation, and it could be mutation rate, population size, and history. But because I kind of tested for that, like I used only like couple genes to limit the mutation rate bias, as well as we know that these have different life histories based on their taxonomy, it kind of calls for another explanation. And so what I think is going on is that they exhibit complex population structure. And so this has been shown in seabirds in previous studies. And what may be happening is even though the seabirds are behaviorally phylopatric. They return back to those natal colonies year after year and are partner phylopatric. They're monogamous, but they, on foraging bouts, and they will visit a neighboring colony and they'll mate with another seabird and then return back to their breeding colony to then lay their egg and raise their chick with their partner. And this has been shown for Laysan albatross on the main Hawaiian Islands. Like if you guys go out to Kaena Point, a lot of those pairs are female-female. And a lot of them are able to do this and be faithful to their female partner because they go and they mate with a different male on another colony. And so this could be one of the explanations of why there's why there's behavioral phylopatry but not genetic phylopatry. And because there's a high genetic diversity and potential moderate gene flow with the endangered band rump storm petrel populations, they may also exhibit this complex population structure. And even though we see moderate gene flow in the band rump storm petrels, in their island archipelagos, there has been genetic structure of the species globally, with most of them clustering by island archipelago. And so this, you can see in this from other research, has shown that the individuals from Galapagos cluster together, so they're mixing within the Galapagos, but they're not flying all the way to Japan, which makes sense. And because the Hawaiian populations didn't cluster with any of those globally that had been previously sampled, it concludes that the Hawaiian populations are also clustering within themselves. And so maybe the band rump storm petrels aren't vulnerable to a genetic, an extinction induced by genetics, but there is other threats out there as we talked about. So predator control and related management and monitoring efforts are still really important to ensure this species survival in the future. And this also, for example, this past summer, I went out to the Big Island colony with help from the Hawaii Audubon Society, and I went to do a nest site selection study. So this is the area where we have those 2 confirmed burrows, and this is what it looks like. It's high elevation, lava terrain, and that crack right here is one of their burrows. So I went out there to measure those burrows and take so like the width and depth, as well as the percent cover of vegetation versus lava versus dirt, to see like which characteristics they might prefer within their nests. And so I went out to the Pohakuloa training area and found there are 2 confirmed burrows as well as potential nests, which were confirmed by either scent, or there was feathers, or by different acoustic evidence. So we're pretty sure there is a seabird there. And then I used a paired sampling design, and I measured other random points around each nest of a lava crack that could have been potentially used by a bird but wasn't. And what I found was that the height of the inner opening of the nest, as well as depth, differed significantly from those random available lava crack openings. And so this likely explains that the band rump storm petrel use of these lava pits with complex passages for protection from predators, because even though there is some predator control out there, it is by no means predator free. And we've seen this by camera trap footage. So this is one of the lava cracks that they use as their nests, and there's a rat right there. And there's also been evidence of birds using burrows and coming in and out, but there's been no chicks present. And all these video evidence, they've also seen those adults being predated on by cats. So there's still a lot more work that we have to do before we start to see these cute little fuzzballs emerge from the nests, because this species is still definitely being predated and is threatened by a lot of different threats. So until we can find their chicks and translocate them, we still have to put a lot of time and effort into finding ways to recover this endangered species. And so I hope that my thesis makes a small contribution to our knowledge of seabirds currently in Hawaii and will help in their conservation in the future, especially within the Van Rump storm petrel. And like Kristen, there was a lot of people that made this possible, my funders as well as partners who supplied my data and samples, and also to the many people in our lab and in the field who helped me. So thank you, and I guess we can take questions now.
Any questions for these fine ladies this evening?
Yes.
I was wondering, do you ever, I guess the word is translocate, but to move like the stilts? We live off Hawaiian Street in Hawaii, and there's the marina and there's the island there on the corner of on the old highway in Hawaiian. And last year we saw stilts and we'd be so excited. Took pictures one day, and all of a sudden they're gone. And we were just wondering if someone had come up and moved them for study or something.
First of all, remind me, did you say they were nesting or you saw them foraging? Or?
They were just there.
They were just there, yeah. Okay.
On the island.
The Pocky Lagoon, right?
On the sandbar on the island. And then sometimes they'd be right in the grass, right there along Hawaiian Street.
Yes, yes. And so actually, something I didn't mention in my talk is that stilts they actually have different nesting and foraging habitat. And so there's a number of places you could see them that they would be foraging. They utilize a lot of different habitats, as I mentioned, like golf courses, treatment plants, things like that. However, their nesting habitat is pretty limited. And so they do only seem to be utilizing, as far as like the core nesting populations, they seem to be utilizing these refuges and these main wetlands that are being managed, as far as our knowledge goes. And so to answer your question, it's very likely that they're moving around. So someone actually, a colleague of mine, is doing a movement study on the Hawaiian Stilt, and to see like how they're utilizing the different wetlands or different habitat areas. So most likely, if you saw them one day and maybe they weren't there another day, that's very likely. So actually, when I go out to do surveys, not during the nesting period, I actually do other waterbird surveys for other reasons. But some days they'll be in Kauai Marsh, and some days, it looks like everybody's over at Hamakua today. So they do utilize and move between the wetlands. So there's definitely not currently any translocation or anything like that, a project that we would move them. But the birds do utilize different habitats, so it's very, very likely that you did see them and didn't see them another day. So, but they may be back. Yeah, but we do need to look. We are trying to look into how they're utilizing the different habitats, because it's really interesting how sometimes they're there and sometimes they're not. So we need to see what characteristics are driving that.
Your Hamakua site. It's a site that's pretty impacted by the human commercial activity, like street runoff.
Yes.
Every data said some really low numbers. So hard to get good data. So it's so linear.
Yes, and so for my first year, my preliminary results definitely had much lower sample sizes, as I wasn't given access to go through the wetland completely to exhaust it and search for all of the nests that were there. So most definitely my sample sizes were an underestimate of the number of nests that were there. And I definitely can verify that because I've already started my nesting season this year, and I've already found way more nests than I did in like some of the total wetlands from the first year. So that was just like preliminary to see what I could find based on the methods that I was allowed to do. But this year, like I said, I will have much higher sample sizes, and so hopefully present again to give you an update on what I find this year. But yes, definitely a lot going on at Hamakua because there's so many different things that could affect them there.
Anyone else? I don't want to monopolize somebody else's question. I was wondering on your bird, do you know the wingspan?
So they're very small. They only fit in the size of your hand. Like I went out actually this summer as well to Kauai and helped with one of their mist netting events. So they have, I don't know the exact number or like the exact by like the top of my head how long it is, but they're one of the smallest seabirds, especially if they're the smallest seabird that nests here, and like their band is like the smallest you can get. It's a 1A, and we didn't actually measure we try to limit the amount of time we have them in our hands. So we try to put the band on, and sometimes we don't get to all the measurements, but it was really cool to see this bird that I'd been working on for 2 years and like in person. Yeah. So they're out there. We just don't know where they're nesting on Kauai. We think that they have a bigger population on Kauai based on song meter data, but we haven't actually pinpointed where the nests are there. We think they're like on really steep like cliffs that you can't get to. Yeah.